operon concept. Jacob and Monod in year 1961 explained induction and repression of enzyme synthesis on the genetic basis or at gene level. They suggested a system for regulation of enzyme synthesis by gene action. Such a system for the regulation of enzyme synthesis is known as operon model, O-P-E-R-O-N, operon model. Operon is a genetic unit which consists of an operator gene, then structural gene, where structural genes are controlled by operator gene for enzyme synthesis. So in this figure one can see the operon, which is a genetic unit and consists of operator gene, see here operator gene, structural gene here, and structural genes are controlled by operator gene for whether to synthesize or not to synthesize enzyme. Operon can be defined as a group of closely linked structural genes and associated genes, namely operator gene, promoter gene, and regulator gene, which controls synthesis of enzymes. Structural genes are responsible for enzyme synthesis. The product of regulator gene inhibits the action of structural genes. The product of regulator gene, see regulator gene here, it is regulator gene, inhibits the action of structural genes. See product here, lack of, this is the product, and it goes and blocks operator gene. And that's why there is no synthesis of these enzymes. So like this, the product of regulator gene inhibits the action of structural genes. Regulator gene is here. And it inhibits formation of these three enzymes. And via operator gene, this thing happen. Inhibition of formation of enzymes. Operator gene thus functions as switch to turn on and off for the activity of structural genes. Means whenever this product of regulator gene, that is lac repressor, a type of protein, it binds with this operator gene and operator gene gets blocked. And that's why there is no synthesis of the enzymes. What is lac apron model? The details we will see now. Like operon given by Jacob and Monod and studied thoroughly in Escherichia coli. Operon is involved in breakdown of lactose in E. coli, and that is why is called as a lac operon. This model is called as a lac operon model. Lac operon in Escherichia coli is a small segment of circular DNA. See here, a small segment of circular DNA molecule. Lac operon consists of 4 types of closely associated genes. So we can see here structural genes, then operator gene, then promoter gene, and near to it is a regulatory gene or regulator gene. Lac operon regulates synthesis of 3 enzymes. We know the names now: beta galactosidase, then beta galactoside permease, and beta galactoside transacetylase. What is the function of these enzymes that we know? Beta galactosidase breaks down lactose into glucose and galactose, while beta galactoside permease facilitates the entry of lactose into the bacterial cell. Beta galactoside acetylase appears in small quantity, but what is the function is not yet known. Lac operon, which is thoroughly studied in E. coli by Jacob and Monod, consists of structural genes, operator gene, promoter gene, regulator gene, repressor, co-repressor, and inducer. Now structural gene. See here, structural gene A, Y, and Z. A, Y, and Z. So these are structural genes. These genes, the direct synthesis of enzymes through their transcribed mRNA. See the process of transcription, and as a result formation of this mRNA. This is mRNA. The lactose operon of E. coli has a small segment of circular DNA. Here it is. This lac operon has 3 structural genes, that is A, Y, and Z, which transcribe only one long mRNA molecule. Here only one long mRNA molecule it is produced, or transcription, after transcription mRNA, single large mRNA is produced. These 3 genes control synthesis of 3 different enzymes, and those enzymes we have named them. So this is what related to the structural genes. Next to structural gene, there is operator gene. Situated near to the structural gene is a operator gene, which controls the structural genes or determines whether or not the structural genes are repressed by repressor. So this is repressor. The gene is closely linked to the structural gene. Promoter gene. Near to structural gene is a promoter gene. mRNA transcription by structural genes takes place in presence of enzyme RNA polymerase. See, this enzyme RNA polymerase is responsible for transcription of mRNA. This enzyme is attached to promoter gene. RNA polymerase is attached to the promoter gene. See here, that enzyme is shown here, over here. It is attached to this promoter gene. Then it moves along operator gene. This enzyme, then it moves along operator gene, reach to the structural gene, and then only the transcription starts and mRNA is produced. The promoter gene controls synthesis of RNA polymerase. This RNA polymerase synthesis is controlled by the promoter gene. Now regulator gene. Near to promoter gene is regulator gene, which directs the synthesis of enzyme and acts as a repressor. It transcribes mRNA. See here, regulator gene transcribes mRNA. Here, formation of mRNA. And this mRNA is responsible for formation of this lac repressor. Now, what is repressor? So, is a protein substance which represses the synthesis of enzymes, and this process is called as a repression. So, when this repressor is produced, it binds with the operator gene, and RNA polymerase cannot reach to the structural gene, which is essential for the transcription. So like this, regulator gene, when it expresses regulator gene, when it transcribe produces mRNA. mRNA produces lac protein, and lac protein binds with the operator, and it blocks the way of RNA polymerase, and RNA polymerase do not reach to the structural genes. Structural genes do not transcribe, and no formation of the three enzymes. Now, co-repressor is a substance which decreases the amount of specific enzyme repressor is activated in presence of co-repressor to form co-repressor complex which blocks the operator. The structural genes therefore cannot synthesize the enzymes in lac operon system. Then inducer is a substance which stops the function of repressor and thus enables structural genes to transcribe and to produce mRNA, and enzyme synthesis starts. Inducer binds to repressor protein. This inducer, it binds to repressor protein, binds with repressor protein and prevents repressor from blocking transcription of mRNA on structural genes. So when this inducer binds with this lac protein, it do not block operator gene. RNA polymerase can pass and reach to the structural genes. Structural genes transcribe and production of enzyme occur. Now mechanism of regulation of enzyme synthesis in lac operon. How enzyme synthesis is regulated? So when inducer is absent, synthesis of enzyme stops. Repressor protein has affinity for operator gene. The repressor protein produced binds to operator gene. When inducer, that is lactose, is absent, repressor protein binds to operator gene and it blocks the path of RNA polymerase. The structural genes are unable to transcribe mRNA and synthesis of all three enzymes, beta-galactosidase, beta-galactoside permease and beta-galactoside transacetylase does not take place. When inducer, that is lactose, is present, repressor protein binds to inducer to form or to it blocks the inducer and enzyme synthesis takes place. As RNA polymerase can reach to the structural gene via operator gene. Now when co-repressor is absent, synthesis of enzymes takes place. When co-repressor is absent, repressor is inactive and it does not bind to operator gene. When repressor does not bind to operator gene, then path of RNA polymerase enzyme is clear, which can reach to structural gene, and synthesis of all three enzymes takes place. When co-repressor is present, synthesis of enzyme does not take place. In presence of co-repressor, the repressor substance binds to co-repressor to form repressor-co-repressor complex. This complex then binds to operator gene and it blocks the path of RNA polymerase enzyme. So the structural genes are unable to transcribe mRNA, and as a result synthesis of enzymes does not occur. So this is how the switch off and switch on or regulatory action on the production of enzymes, or call it as a proteins, takes place. Remember here one thing, all enzymes are proteins, but all proteins are not enzymes.